Okay, so first of all, good afternoon, and thanks a lot for the chance to present here what we are doing in OmicsDI. To this audience, I don't really need to talk about the motivation, but really we wanted to develop from the beginning something which for omics data sets allows to do the same thing which people are doing all the time with PubMed or EuroPubMed Central nowadays. You can go to one central entry point and you're fairly sure you find the relevant publications. For omics data sets, at least in the last few years, there has been increasing collaboration between the repositories in each of the omics domains. And you can now go to Proteome Central to centrally search all the proteomics repositories, to Metabolome Central to search the metabolomics repositories where they are participating in metabolomics change. But really, there's not yet the central entry point. And so the vision is really to create a kind of PubMed for omics data sets. And I think we are all sharing this vision in a much larger scale here for many people, for almost everybody in the room, beyond omics. But for us, it's really the target is the more restricted area of omics. And this is where we are at the moment. You can access the site. What is later? This is later. So OmicsDI.org is working, I hope. And you can access it and see where we are at the moment. We have a graphical browsing entry page, which allows you to access subsets of what we have indexed so far based on a number of attributes, the word cloud. You can browse by tissue, organism, or diseases based on the metadata we are indexing through this graphical widget. You can browse by repository so that you can easily see without even knowing the relevant accession numbers. Of course, we also give examples, but you can easily get into OmicsDI to see what's really there. And this is the current status in terms of numbers. We are indexing 11 databases from 3 continents with altogether 78,000 data sets. And these come from proteomics, metabolomics, transcriptomics. And we also include EGA as the only resource which is protected access. EGA, the European Genotype Archive, is the European equivalent of dbGaP. And so we can also test how does it work if you have only the metadata accessible, but not the data itself. A key point of interest is access metrics. And so one thing we are providing is a direct count of accesses to the different data sets right on the home page so that you can see where there is heavy access in hot data sets at the moment. This is frequently updated. We provide the latest data sets, so really to make it interesting, inviting to interact with new data sets to see what's around there, but we also provide statistics overall across what is the contribution of the different repositories over time, of the different data types over time. And this is the search results overview you get when you use the search box. And I quickly updated the slides. You can probably not see this here, but you all received an email inviting to participate in testing of use cases for repositories. And the example which is mentioned in this, the email was sent 2 days ago, is search for human glioma involving IDH1, IDH2 data sets. And so I just updated it to this. And so this is what you get. You get an overview of the identified data sets. I'm quite happy because all the identified data sets seem relevant at first glance, but not everything is perfect. You can't see this here, but the the search refinement offers options for refining to mus musculus or human. So actually it did identify the right species for the data set, but although human was in the search terms, that is not yet taken into account. But at least all the data sets which we provide look relevant already. And then you can refine to use only the human data sets. And then you can drill into each of these data sets. And you get the data set specific view. This again doesn't look very different from DataNet. Both of these went through a kind of co-evolution. I'll come back to this in the last slide. We provide a minimum set of metadata to characterize the data set. And we've done quite a lot of effort in providing one of the key features, which is now making PubMed successful, which is related data sets. So what else is out there which might be relevant for you if you came across this data set, perhaps even with a very specific search with a PubMed ID, but what else is there? And so I'm going to mention 4 different types of related data set options which we are providing. The first one is the more or less obvious one, related by metadata. So for each data set, we show related ones which are dug up by a similar principle like Google does. These are the other data sets which might be relevant to you based on the metadata. To do this, we are also using ontology-aware indexing. We are using the ontology tools from NCBO to figure out relevant ontologies and relevant terms in the metadata and try to enrich our indexing by these tools so that you find also more than just by pure text, full text search. And one of the relationships we use is by publication. At the moment, when you have a multi-omics study and you submit the data to the relevant repositories, that means very often basically splitting up the deposition and sending the metabolomics data to for example, metabolites at the EBI or the metabolomics workbench, the proteomics component to PRIDE or Peptide Atlas, the transcriptomics component to ArrayExpress or GEO, and often it's quite difficult to put these pieces together again. So we are indexing the common publication, which is referenced in all the data sets which are stored in the different repositories, so that we can directly show this widget here, this multicolor widget shows this is actually a data set which belongs to a multi-omics experiment. And then you see in the data set the pointers to the other, to both the original data set. So this data set you see here is actually a proteomics data set stored in PRIDE. But you also see other related omics data sets, and in this case a pointer to ArrayExpress because related data was deposited in ArrayExpress. So by the common publication, but then also tracing accession numbers, we are establishing where do data sets which are stored independently really belong together because they have all been generated by the same, or they are all supporting the same underlying publication. The next point, and again something which is very interesting not only from the scientific but also from the funding point of view, is the relation by data reuse. And more, the idea of open data is not only to allow scientists to validate the conclusion of the individual publication, but also to use the data and to query this data with a new question, perhaps across many different data sets. And so and then of course, the idea is that the original data set is properly cited, that you can make the chain from the derived new interpretation of the data back to the original data set. And we want to support this as much as we can in the OmicsDI by collecting this data from the participating repositories and tracing it and making it easily visible. So where we have the data, we are showing not only the link to the original data set, which you came across, but also reanalyzed by. And so in this case, there's a pride data set which has been reanalyzed by GPMDB, and you directly, one click away, have both these, the original data and the reanalyzed data in another entity. And in some cases, this can be fairly extreme. Like in this case, this is the data set which supports one of the two draft human proteome maps, and this extended over many different tissues and samples, and each of these has been reanalyzed by Peptide Atlas as a different new reinterpreted data set. So here you have a whole bunch of reinterpreted data sets. All of these are also accessible through OmicsDI, but here you have the whole link of all those data sets which have been derived from this one source data set. We also provide, of course, the connecting publication, the publication which belongs to the data set, where it is there, which is mostly there, and we connect this to altmetrics so that you can also see an alternative view of what was the impact of this data set through altmetrics, and you can also comment on the data set, again a feature which you've already seen previously today. Technically, again, a lot of this will sound very similar to what you've heard already. So we are collecting the metadata from all the participant repositories, and we are collecting it in a very simple OmicsDI XML format. And then this is pre-processed a bit, and then it goes into the EBI search indexer. So in the development of OmicsDI, we have built on existing infrastructure, which is already at the EBI, and which is already indexing 1.1 billion entries at the EBI internally. So we are fairly sure that it will scale, and it has scaled quite well, going from 8,000 proteomics and metabolomics data sets to currently 80,000. And then we are using the web services provided by this EBI system, which was pre-existing, to then build the OmicsDI website through the OmicsDI application. This looks very similar to what Susanna presented just a bit earlier, which is a good thing because it made the mapping fairly easy. So we have, the only difference is we have three categories of attributes: mandatory fields, desired fields, and additional fields. And Susanna already mentioned, as we are focusing on omics, there are some specific areas which are very specific to the individual domain, and these are only relevant in this domain, so these would clearly be additional fields. Mandatory and desired are more the common elements. I've already I won't go into the technical detail here. The only thing is the existing infrastructure we built on is not a database, it's an indexing system. That meant we had to provide our own database to provide those things which are specific to OmicsDI. This is some parts of the data pre-processing, but also the statistics we collect. So if we collect click statistics, then clearly we have to store them somewhere. So this is an added level on top of the pure indexing system. The indexing system is probably very similar to DataMet in the sense we are building on publication, on metadata. Mainly publication is the one which allows cross connections between different domains: proteomics, genomics, metabolomics, but also other metadata attributes. But the key point for omics is we have another anchor. We have the molecules. In each of the omics data sets, we have a set of molecules which are somehow relevant to that data set. For proteomics, these are normally all the observed proteins, because in proteomics there's still an undersampling. So you will always only have a subset of all observable proteins in your result set, although the coverage has rapidly been increasing in the last few years. But so we are now asking the repositories also for the list of the identified or quantified proteins in each data set. And the same thing for genomics or metabolomics. We ask the repositories for the list of relevant molecules in these data sets. And of course, in these domains, this won't be just the observed, at least in genomics, it's not the observed molecules, but it's those which will have a significant fold change or something like this. And we map all of this then to reference databases, UniProt or Ensemble or Campbell or Caddy, to be able to make cross connections between the different data sets. And this really allows us to do something nice like this. We have another relationship type between data sets, completely independent of the metadata, and that is by shared molecules. This chord diagram shows for the starting point data set, this one here marked in red, it shows a number of closely related data sets only by their content in terms of molecules, and it shows graphically what percentage of molecules are shared between these different data sets. Sometimes these chord diagrams are very dense. That's why you have a threshold slider where you can change the cutoff so that you can make it clearer or that you can really see everything. And then you also see the same representation, the textual representation of these data sets here. So this really allows us a completely new dimension. Even if somebody has provided no or practically no metadata, you can still find related data sets by the actual data, by the molecular content of these data sets. Of course, we need to have at least these molecules, but very often you can even derive them from the data set if you want, although that's, of course, not desirable, but we should get them provided by the repository providers. And so this is the fourth and last type of relationship between data sets. So we have standard metadata attributes, publication, reuse of data, and shared molecule sets as the relationship types between data sets. And this last category also opens an interesting perspective for the future. So far, these relationships only happen within each domain. So we only show proteomics data sets related to each other, metabolomics data sets related to each other. But of course, you can either, at least between genomics and proteomics, we could establish a mapping and then really establish connections between proteomics and, let's say, transcriptomics data sets. But there's also another very interesting use case, which is in the area of pathways and systems biology. A pathway can be simplified as an entity, a gene set, combined with a set of metabolites. And so we could link different data sets via common pathways. This is for now purely research. This is not something which will easily move into the production version. But it should be possible to establish non-obvious connections via pathways, or in the similar sense, via systems biology models, which again are entities which link sets of genes with sets of metabolites or other molecules. And so we are starting to look into how we can establish cross connections between data sets in these ways. And with that, back to what has been mentioned before here as the ecosystem. A lot of this presentation you will probably have heard today in a similar form in one of the other related projects. And the relationship between OmicsDI and BioCaddy, Susanna already mentioned this. Originally, these were independently funded, but we have had co-evolution with lots of cross-connectivity, gene exchange, which was then also officially sanctioned with an administrative supplement, where a number of the PIs are working together to really establish compatibility between OmicsDI and BioCaddy. Susanna already described the mapping from OmicsDI to BioCaddy. The next steps, of course, are then to provide the actual data which we collect for OmicsDI to BioCaddy. And there are other elements in this joint project. For example, generalizing the visualization widgets we have developed. And there's also some work on joint access metrics between the two. And the idea is, in terms of long-term evolution, OmicsDI is aiming to go more into the depth for omics, and I've shown how we are using the specific characteristics of omics. And for BioCaddy, which has a much broader coverage to go into the breadth, but of course, we want to have the same data accessible in BioCaddy when, let's say, refinement work is already being done for OmicsDI. And with that, I would like to very specifically acknowledge the two people who really have made this possible. Yasser Perez-Riverol and Ming Zabai are the two main developers of OmicsDI and have really done a great work. They were supported by many teams in all the resources which provide data, and by the EBI search interface team, which has already built the EBI search indexer. And of course, all of this work wouldn't have been possible without being involved in the UCLA BD2K Center of Excellence, coordinated by Pei Ping. And of course, thanks a lot for the NIH funding which made this work possible. Thank you.